I'm gonna share screen here, and I think can everybody see my screen?
Yes, we can see.
Yes.
Do I have a yes?
Yes, it is.
Okay. So so what I'll do is, if everybody remembers, well, for those who were around at the meeting just over a year ago, we talked about a plan to start integrating and leveraging some of the expertise that exists within the Global Brain Consortium to develop some strategies and some products that we can use to leverage to do a little bit what we talked about in the last call, and that is kind of take some of these large data efforts and make them more broad. So over the past year, we've been working on that, and I'll talk about that a little bit today before we move on and have a panel discussion. So this is the GLAD project. We think it's a very unique opportunity to leverage this expertise to build big data sets. I, as in charge, partly in charge of this, was kind of needed to identify someone who kind of take the lead. So a lot of what I'll be talking about today has actually been driven in large part by a postdoc, hopefully soon assistant professor Emmanuel Bani. Unfortunately, he won't. He did his education in Uganda. Unfortunately, he won't be able to join us today. He's at the SRCD meeting, but I'll want to give him his due, and then I'll talk about some of the progress we've made over the last year. Okay, so just a brief overview. I'll talk a bit about just a bit of what we talked about last year, an overview of large-scale brain research and studies that are currently gone, went on some challenges with regard to making them kind of more inclusive, and then a bit about our new white paper that's nearly complete. It's also put together as a call to action for doing more representative worldwide developmental brain health study. All right, so there are now, I'd say, several instances or several which has been very fruitful works out there about large-scale data initiatives. I think probably this kicked off with the Human Connectome Project, which is collecting in the U.S. from just 2 different sites, the Washington University and the University of Minnesota, about 1,200 participants. Since then, there have been several large initiatives, including including the Biobank Human Brain Project, and those like the ABCD study, the Adolescent Brain and Cognitive Development Study, which is the U.S. national study of 10,000 youth starting at the age of 9 and 10 years old, and following those youth longitudinally with both imaging data, psychometrics, and behavioral assessments, clinical assessments for essentially 10 years. That study is about in year 7. I'm part of the data core and one of the PIs of one of the data collection sites, and that study has been very fruitful, but it suffers from large, important representation worldwide. New initiatives such as the Healthy Brain and Child Development Study is going to follow up with some of these types of longitudinal development studies, is repeating, but including EEG and some other biospecimens and other types of assessments, and starting at the neonatal period, or essentially in the 2nd trimester, and following those kids until they're about 10 years old, but follows the same general theme. The issue is whether from the China Brain Project or all these ones that I've been talking about, UK Biobank, is they're not really representative of our world community, and that provides some inequities and some challenges for improving our ability to have more representative samples for the science and the applied work we all do. There are several challenges here on that front. One is that there's sampling bias, various sampling biases, measurement biases. We have what we call normativity biases, how people generate theories about about brain health, brain development, mental health. And there's just a large empirical gap in our understanding of how various cultural environments interact with mind, brain, and behavior. So the GLAD initiative set off with a few main goals. One is that we wanted to foster the current collaborations to improve brain health and support service providers in low-income countries. One, we wanted to develop a strategy of inclusion for big data. Mike just got done talking about a lot of these big data initiatives and what he's learned from this and how important they are for our understanding. But that leverages these collaborations and then starts developing new ones to go beyond. And then 3 was to provide a roadmap to engender the important support and resources to kind of kick off some of these types of initiatives. Okay, so where are we right now? So as I said, in November of 2021 is when we kind of kicked off this effort. We pulled together several individuals at my institute in Minnesota called the Masonic Institute for the Developing Brain. We have a global health service unit. The Emmanuel, who I introduced earlier, kind of took the lead of this efforts to kind of pull together some administrative support and our, the collaborators to start building a model of what we need to do to start generating some support. In January through May, we added a few additional collaborators, and then built kind of a framework of how we might want to think through developing our ideas. From there, we leveraged the curiosity and intellect and minds of the Global Brain Consortium members. They're in the GLAD project to start fleshing out those ideas. We, in essence, developed 2 specific working groups. One that thought about to think through like a design of something like this, where you could do a large-scale study across multiple countries. And then the other on the informatics piece with a heavy emphasis on leveraging current expertise, particularly with Alan's group at McGill, but also assuring that whatever data repository or setup that could be put together, that it was accessible by everybody and leverageable by everybody, regardless of whatever circumstance they're in, as long as they have internet. And from there, after those few meetings, we began preparation of a white paper, started getting some feedback, making some revisions, and we're essentially at the final stages of the revisions of that white paper to be submitted. Okay, there's some big takeaways from the effort so far. One is that there are several existing resources that exist that exist among current GBC members and others. That the type of evaluation and what we need to do for harmonization for data across in this unique context can be done. That this globalization, what we call globalization of brain health, that it is extremely needed, and that overall this whole GLAD initiative is doable. We kind of broke this down into a couple of phases. One is that there are certainly several large existing data sets that exist across the Global Brain Consortium members and others that probably would want to start collating and harmonizing this existing data for this purpose in the beginning. Another phase on design, the prospective data collection with our partners, leveraging common elements along with the site-specific expertise, and then phase 3 would then be to begin this worldwide collaboration on developmental brain health. Here's the white paper, what Emmanuel is calling is the living blueprint. You can see in green, we have a lot of interesting existing cohorts of folks that would be interested in already contributing. There were several potential cohorts from other locations that were identified that could be leveraged in some of these initial phases. And there were also several suggestions where we still need to reach out and generate some partnerships for many other additional cohorts to start building out a global repository. Okay, so there are several critical components of the white paper that were put together on pretty much it's kind of identifying the problem, characterizing the challenges, and then kind of outlining some of the solutions. So one is that there's some critical gaps in infant, child, and adolescent research across the globe, particularly around proper representation, diversity with regard to methods. We talked about, which includes aspects of various cultures, and the relevance of certain types of data in specific contexts. There's lots of promise of large-scale multi-site brain kind of projects of this type and beyond. I think Mike gave a lot of examples from a lot of his work in the earlier talks today on how this has been leveraged in the kind of Western kind of context. We think, but there's some additional benefits of doing this worldwide. One is this foster this international collaboration, can be much more locally responsive and international, internationally relevant, and then it provides data and knowledge sharing across groups as we diversify essentially the ideas and the thoughts into the work, that the chances of us coming out ahead more quickly and accelerate our progress is much more attainable. We add an aspect of the GLAD initiative itself, what the priorities of the initiative are, the innovative methodologies, the aspects of some of the broader samples, the behavioral neuroimaging, what can be done today, what can be done in the future, and that includes differences between things that are more accessible like EEG as opposed to kind of expensive, more one-off things like MRI, who potential collaborators could be, and so on and so forth. And then there's another aspect of the clinical and public health applications, which are quite large, right? And that's things like developmental charts, useful detection for identifying disabilities, brain markers for interventions, policy documents. The list can go on as far as the implications and the applications towards clinical and public health. Okay, so I'm gonna end there before we can have a little short discussion, maybe 5 to 10 minutes, before we go to the panel. But the conclusion is that large-scale discovery science provides unique opportunities to advance our work and foster population brain health for everybody. Strong partnerships with communities, researchers, and funding bodies and organizations such as WHO are going to be critical for our ability to launch such an effort. And that this certain level of humility is required from all of us to make sure we develop an inclusive framework that can be implemented and promoted for everybody. There are a few next steps that are currently in the works. One is we're finalizing we want to finalize and publish this white paper, which again should be done and coming out very soon. Well, we are beginning to start to plan and beginning to engage strategic agencies for funding and support. And then the last bit is it will be important that we expand our network of collaborators to do this in earnest. So I'll stop there. We have just a few minutes for some discussion, and I'm hoping that we can have a lively interchange here on what people think and what folks think we should do next. Thank you.
Okay, anyone, if you have any questions, you can ask Professor.
Okay, I'm moving the chat here. All right. Thoughts? Questions? Yes, Sid.
Yeah, Damien, thank you. As you know, I've been working with,
Oh, is Sid muted?
I'm not muted.
Oh, I can hear you now. Yep.
Okay, good. I've been working with Pedro on trying to work out an EEG common protocol that would work and for all purposes, and of course that's impossible. And I'm gathering ideas from various places. I just learned that HBCD project is now moving from MRI to also include EEG. They haven't started yet. I haven't gotten
We have started.
They have started. Can
Yep.
you send me the protocol so I can compare with what I've been getting to try to put it all together to try to
I
see if we
can. Yep. There's a standardized protocol for that will hopefully work across ages. But yeah, I
Fantastic.
absolutely can do that. Yep.
Okay, please do that, and then I'll pull it together for the white paper. We can pull in all the details.
Absolutely.
Thank you.
Any other thoughts? What I can tell you is that, I think one of the more obvious conclusions that it'll take quite a bit of time for design and kind of generating some of these common elements. One of which may be EEG, but the behavioral clinical elements, the design aspect of this is going to be challenging. We identified a few, but that will take a lot of deep thinking to think about how, to think through how that will work. But that will take some time. It will require some resources. That was definitely one of the pieces that we learned. We talked a lot about what age, because that was another big topic. So we had a lot of discussions on the age groups, and we don't think that it's probably feasible to do something similar to what we did for what's been done for studies like HBCD or ABCD, where you do longitudinal sampling starting at one age and just kind of follow on. It will have to be some type of lag design or something like that for the prospective part of this. And I think that we believe that initial phase 1 will help us tell us a lot about what that might look like. There's a lot of other aspects about, using aspects of data science to help let the data tell you how things harmonize instead of trying to do a lot of that upfront for some of these for some types of measures. And again, some of that initial phase 1 would allow for some of that exploration. Trying to think of some of the other big topics. There's a bunch. MRI was a big one, because it's so rich and being leveraged in a lot of these big studies, but it's certainly not going to be accessible or feasible to do that everywhere. So in the design of such an effort, almost certainly there'll be outside of the common elements, we probably likely encourage, shared measurements that are unique to sites or that are maybe a subset of sites, would run, and the MRI would likely be one of those. Even EEG in some cases, probably. I can say, and this will be talked about the next, probably likely over the panel, but I was very encouraged by the efforts by the group as far as the current data strategies and some of the studies that are going on. I know I don't see him on here, but I know Faisal and their group have a really amazing data resource that's being put together that I'm sure that will be talked about later. That maybe definitely would likely be a good program to look at as the design starts to build out. But there are several others as well.
Okay. Thank you so much, Professor Damien, for your wonderful talk and for everyone's questions. I would like to say a few things. First of all, I would like to introduce myself. I'm Sherry. your moderator for today's session. I am in Pedro's lab, and I welcome you all here. So just a few things. One thing is that please, it's a humble request to like respect the time limit of your presentation, as we have you want to do at the end of your presentation. You can manage your time accordingly. And if you want to do it at the end of our session, we can also do that. So now I would like to give the platform to Professor Janina Gallagher, who is going to talk on the neuroscience of early adversity and will further take the charge of our panel. So thank you. Professor, over to you.
Okay, so I'm just going to very quickly go through a brief introduction of the Barbados Nutrition Study. And just to let you know that we have, 5 speakers this morning. Everybody's going to try to speak very quickly. And we're going to summarize our updates really in the last year and a half on the study. So again, just to put this into context, childhood malnutrition continues to be highly prevalent across the world, particularly in sub-Saharan Africa and Southeastern Asia. But it also exists in the Caribbean and in South America. And we have, which is where our focus predominantly has been. These figures show you that even though there was a goal, a millennial goal to exterminate, to get rid of undernutrition in children under the age of 5, we continue to have 20.8% roughly of the world's population. And actually with COVID, that figure has actually increased.
Excuse me, Professor,
Yes.
sorry to interrupt you. Can you do your full screen for the PPT? Thank you.
And again, the only thing I talked about before really were the prevalence, figures. And again, it's probably about 23% of the world's population of children under the age of 5 that continue to be exposed to undernutrition globally. So it's a very significant public health problem. The Barbados Nutrition Study, which started in 1967 and is now in its 56th year, has been tracking individuals with exposed to protein energy malnutrition limited to the first year of life, never had an episode thereafter, as we have tracked and followed these people carefully throughout their lifetimes. And also, can you see my pointer here as well?
Yes.
Okay, great. And there is in parallel with our malnourished cohort, we have also been tracking controls who were healthy classmate controls, who were matched by age, gender, and handedness to the protein energy malnutrition cohort, and our neighborhood children effectively. The selection criteria were that all children were born between 1967 and 1972. They had birth weights of over 5 pounds, over 2,500 grams. They had good APGAR scores. They all had prenatal care, no birth complications, and no childhood encephalopathic events. None of them, as I mentioned before, have had any undernutrition beyond age 1 in life. So this effectively shows you in red where the malnutrition exposure occurred. It occurred postnatally for the first year of life, and the children had moderate to severe malnutrition at this period of time, but they had adequate nutrition throughout their lifespans. And we have 312 in the G1 population. We also have their parents and data on their parents. So, and the generation 2 data, which is actually greater than 206 at this point, brings us to 830 individuals, not counting the additional G2s that, that have been underway since 2018 and who we have not tested as yet. So we have a substantial sample, that can be tapped into for future studies. Okay, now I'm having trouble advancing the slide presentation, even though I was able to get it on this particular screen. So I'm not sure how to do this.
Press the right-hand arrow on your keyboard.
It is not working.
Is there an arrow on your screen? If you can see it, you can hit it. It should work.
If what? Pardon?
Can you see an arrows below your slide?
No, there are no arrows. it's a full slide. So I have to stop again.
You don't have any presentation mode open on another screen?
I do,
Okay.
but it's not working. It isn't working.
Okay.
So
Okay, Professor, if it's not working, would you like me to share my screen and I can share the slides and you can do the presentation?
Sure, but how can you do that?
I have your PPT with me.
I don't think you do.
Okay, let me see.
It's been changed. It's been changed.
Oh,
Yeah, I'm so sorry for the technical.
Okay,
let me stop
now
sharing
oh,
again.
now it was
Just-
working.
your screen was working now. Just before you unshared it, it was working. The slides were moving.
Yeah.
No, but
Yeah,
I'm
can you-
not. Go ahead. I'm not able to back up and get to the earlier slides.
Oh,
I've missed many slides here. So I don't know how to handle this. Let me just verbally try to summarize for you and I'll just move forward. Basically, we've had 7 waves of data collection for the Barbados nutrition study, which start in 1967. We had a very comprehensive phase of data collection when the children entered school in 1977 and 1978, at which time we collected EEG data. This was collaboratively with Eroy John and the Cuban Neuroscience Center. And as I mentioned, there are 7 waves. The most recent one was between 2018 and 2020, when we had a collaborative study together with the Cuban Neuroscience Center, with University of Montreal, and Gallagher, and you know, we're able to collect data on at ages 45 to 51. Now, the key findings, there have been multiple findings, both with respect to QEEG findings, as well as epigenetic findings and behavioral and cognitive findings. This is a multidisciplinary study across multiple venues. And today we're going to be focusing predominantly on the final phase and the earliest phases from the longitudinal perspective, and also on a very significant finding, which is the MoCA and MMSE findings of accelerated cognitive decline, which we documented when the individuals were 45 to 51 years of age. Now, let me see if I can advance again. I can't, as you can see, right? Is it advancing? No.
yeah. No, it's not.
Okay. So I think, you know, with that, you know, just giving you a little bit of a background on the study, I also wanted to, you know, move on to present my collaborators, you know, who will each be giving you an overview and of, you know, their particular aspects of this work. In the past year, we have published and submitted 6 publications, and there are several others that are very close to being submitted. So we've made a lot of progress in that regard. We've also made a lot of progress from the point of view of engaging at this point collaborators, but also a potential reliability sample in Jamaica, where a parallel study looking at early childhood malnutrition and long-term consequences has also been in effect. And, you know, we will be using this as a reliability test in the future. I know we talked about that. That was discussed earlier this morning. In any event, at this point, I'd like to present Dr. Jorge Bosch-Bayard, who will begin with the first summary of his presentation. Thank you.
Hello, Janina, thank you. Hello, everybody. Let me see if I can share my screen. So inside this huge project that Janina has been presenting, what I'm going to present is the result of the EEG comparison of the longitudinal sample This is sorry. Okay. So this is a work that we did in which we compare the EEG recorded in 1978 at the childhood, at the onset of the study, and the EEG recorded in 2018, more than 45 years after. So these children that suffer malnutrition, pain malnutrition, protein energy malnutrition during the first year of life, they were between 5 and 11 years old in 1978 when the first EEG was taken, and they were around 60 for more than 45 years old when the second study where EEG was recorded, the one that we are analyzing today. So this is the description of the sample. Janina already mentioned all the inclusion criteria. She mentioned many of the characteristics of the sample. I am only going to say that from this very huge study, we were able at this point to record 118, to restore 118 recording from 1978. And in 2018, we were able to get 94 subjects. This is a distribution, the pain and the control, 46, 52, 43, and 51. And 23 pain person have the 2 studies, and 30 person have the control person have the 2 studies. So not all the subjects have the 2 studies. What we did was that we applied the quantitative tomographic EEG analysis, the QEEGT methodology developed at the Cuban Neuroscience Center. We took the EEG of each individual, around 24 epochs of artifact-free EEG, and we analyzed 2.67 of 2.56 seconds. We calculated all the so the average reference, all the methodology. We obtained the EEG spectra. We applied a global scale factor, that is a factor that we developed for accounting for variability not related to neurophysiology, so it makes the recordings more comparable. Then we applied the Vareta technique to obtain the spectra at the sources, and at the end we obtained as the output the EEG spectra at the 19 channels at the scalp and 3,244 sources at the EEG generators in the frequency range from 0.39 to 19 hertz with a beam frequency of 0.39 hertz. So the next step for this was applying the statistical methodology. For this, what we did was to apply a linear mixed effect model in which we include the group, the study, the interaction between the group and the study, and a random factor of the subject to account for the subject who has the 2 studies. And we calculated the maturational effect for the control and the pain group. This means the EEG at 2018 minus the EEG at 1978. The dysfunctional or the comparison inside each group between adulthood and childhood. So in childhood, it was pain minus control in the 1978, pain minus control in 2018. And also we took a more complex contrast, that is taking the difference between the 2 groups, but taking into account at 2018, but taking into account the starting point, so the differences that they had in 1978. I am going to present only very briefly the result of this. I think the 3 more significant results. But before that, I have to say that having such an incomplete sample, we had to make a very huge work that was mainly done by Pedro and Fuleah A Razzaq to demonstrate that the effect of missing the missing data was did not invalidate our results. So they did a complex analysis in which it was demonstrated that all the subjects that had been missed was at random, and that the statistical analysis still have validity under these conditions. For the pain group, the malnutritional effect, so this is comparing adulthood minus childhood. We found that in the adulthood we had, in the childhood we had more delta activity, that significantly more delta activity than in the adulthood, and we found in the low alpha we found a very significant differences between childhood and adulthood. So in adulthood they had more low alpha than in childhood. I want to explain here the colors. Means we are only taking into account here the results that were significant at P0.01, that either in the positive or the negative, and these two, the yellow and the light blue, are significant at 0.05. All of these results were obtained by applying permutation technique to account for the multi-comparisons. And I forgot to say that we are not using here the raw EEG spectra. What we are using here are the fifth cohorts that we obtained from the comparison with the Cuban normative databases. So it means that we first eliminate the or correct by the age effect. So what we the result in the delta band means that the children during childhood they had more slow brain than in the adulthood, and in the low alpha they exhibited a greater activity. I'm going to explain this later. When we compare the adulthood, the result at the adulthood, we didn't find any differences in the delta and the theta band. So it means that the group, the pain group, improved. So they improved the activity in the delta band. So it was corrected by during the age. They didn't exhibit a pathological higher activity in the delta band, but they exhibit. So this is adulthood. So this is pain minus control in this case. So this excess in alpha means that the pain group has more activity in low alpha than the control group, and the control group has more activity in high alpha than the pain group. We interpret this result as that the maturation of the EEG for the pain group was not complete. So in fact, the mean alpha peak, the average alpha peak for the pain group was about 8 Hz, while the alpha peak in the control group was around the 10 Hz. So it is showing that the pain group, even that they tried, so they had an evolution in the EEG, they didn't reach a full maturation of the EEG. The EEG was always slower than the pain group. And this excess, so they show also an excess in the beta band compared to the control group. This another contrast is, you can see they are very similar. This is the result of the comparison in the adulthood between pain and control, but taking into account the state they have at the onset of the study. So this is a complex contrast in which is pain minus control, but also post minus pre. So taking into account or correcting the analysis by the state at the onset, and this shows that the differences are very similar. No, the interpretation that we do to this is that this excess of low alpha, as I already said, indicates a pattern of different pattern of maturation of the control of the pain group that didn't reach, were not able to obtain the same maturation as the control group, and the excess of beta in the pain group is also related to, has been also related to premature aging or cognitive decline. This has been published by other authors, and you saw that Janina showed also that this is consistent with the finding we have with the MOCA and the mini-mental test. So in general, the result after the result of the EEG comparison after 45 years show that the pain group have a tendency to have a premature aging and a cognitive decline, and this is consistent with the rest of the other measurements that have been analyzed in this group. So we found that the quantitative electroencephalography tomography can be a valid tool to measure the effects at long term of early malnutrition in the brain. So I will stop here because we have many speakers, and I think we.
Okay, thank you so much, Professor. If you have any questions, you can ask, or you can put it in chat, and Professor Bosch can try to answer them.
Or we can. Okay. Yes.
May I? Bosch, very interesting. Perhaps I missed it, but did you say to what extent this would be diagnostic at the individual level? That is, do the distributions for those who have been had experienced serious malnutrition, does that distribution not overlap with the controls?
No, the control did not suffer any type of malnutrition during the suggestion. Yes. This was very well. They were very well selected. They were.
But that, no, my question is. So my question is: did the EEG was the EEG different enough that you could use it as a diagnostic, or is it that the distribution simply were as a group statistically different? Related to the question to the discussion we heard with Michael.
If your question is if we can use the EEG as a diagnostic for malnutrition?
Where the distribution is so different that you could do that. No.
So I don't think that we can use the EEG itself to make a diagnosis, and this should be more appropriate for a biomarkers analysis. So if we find an equation regression that classifies between control and pain, so we can say which is the classification power of the EEG to do this. But in this analysis, we only find significant differences. We don't find which is the classification power of the EEG to differentiate between the two groups.
Okay, thank you. That's all I wanted to know.
Mm-hmm.
Next up, we have Dr. Fuleah from Pedro's lab, and she has shared her PPT with us, so we can listen to her presentation. I'll try to share my screen.
So basically, the variables we are using for this one is the EEG, qualitative scale total EEG scores of neurological science from both the first wave, the baseline, and then the most recent one in 2018. And we are using the cognitive measure MoCA from the 2018, and potential confounding variables are age, gender, and childhood standard of living. So here Bosch already explained that you can see the clear differences between the EEG spectra. It's very clear that the PEM here the EEG is very dispersed, whereas for the 2018, you can see the alpha peak is in low alpha side in comparison to control. And then here is basically the mediation model we are looking into. And you can see that a brain state that we are trying to measure is basically composed of EEG, GT, and soft neurological science. So it's a latent variable. And then cognition also is a linear combination of the MoCA subscores. And we are doing some dimensionality reduction and then doing latent variable modeling to reconstruct these variables. These are the results from some association-based dimensionality reduction where we are just trying to capture the most relevant variables which are related to association. The malnutrition status is not part of this whole process. We're just trying to find the association between the cognition and the EEG and GT and soft neurological science, and we're picking on the most relevant variable to reconstruct the brain state. And then these are the final two mediation models that we have tested. So the first one here, the figure A, that is the mediator here is the brain state from 78, childhood, and then the outcome variable is the cognition. And we saw that the average causal mediation effect was highly significant and in negative direction. Whereas the direct effect was not significant, and this was kind of full mediation here. And then this is the result from the sensitivity analysis for the unmeasured confounding or sequential ignorability. And you can see that the sensitivity parameter for the whole range of the parameter, you don't see any changes in the direction of the original mediation effect. So there's so it's not becoming zero or switching to positive. So it's always negative, which shows highly robust results. And the second mediation model here is actually when you have the main mediator here is the brain state from 2018, the adult life, but there is a second mediator in the model, which is the brain state from 78. And you can see that there is a link that the brain state 78 is actually connected to 2018. And when we do an independent mediation just for brain state 2018, of course, that was significant that the brain was explaining the cognition. But as soon as you put the second mediator, which is the 78 brain state, you see that the average causal mediation effect becomes zero. Which tells us that the childhood brain state or the brain state at the school age is actually explaining away all the cognitive declines or whatever long-term symptoms the subjects have due to malnutrition. And sorry for
Hello,
this.
Fuleah. Sorry to interrupt you. You have 2 minutes left for your talk. Thank you.
No, it's okay. I thought we are running short on time, so maybe we can spend that on question answers.
Okay, but I think it's better if we can do the question answer session at the end, because we are almost
You can
catching
start.
up. Yeah.
Okay, we can start the next talk. Thank you.
Thank you so much. You can stop sharing your screen.
Okay.
Okay, Professor Janina, you can take over and invite the next participant.
Our next speaker is Dr. Anne Gallagher, University of Montreal.
Hi, thanks, Janina. Can you hear me?
Yes, perfectly. Thank you.
Okay, so I'm going to share. Hopefully it will work. Okay, thanks. So I'll try to make it quick. First, I'm really happy being here at the GBC meeting and see you all, guys. I'm gonna present the EG-FNIS part of this collaboration that Janina presented earlier. So this work has been done with, of course, with Janina, Pedro, Maria, Jorge. And I have to say that this the EG-FNIS part of the project is led by Cassandra Roger, who you can see here. She's also with us, I think. And she's a PhD candidate in my lab, and she's the one who went to Barbados in summer 2018 with Janina and other people from the collaborative work. So, okay, so it seems to work. So we acquired, for this part of the project, we acquired data on 55 participants from the original BNS cohort. 26 of them were previously malnourished adults, and 29 were control participants. They were all age, like Jorge said, between 46 and 51 years old when we saw them and when we recorded EG-FNIRS. For the FNIRS part, we used the NeurScout system with 16 sources and 16 detectors, yielding to 46 channels. So you cannot see it very well here, but that's the cap we use to all the EEG electrodes and FNIRS optods. So all the fibers were distributed on the scalp on the frontal, temporal, and parietal areas. For the EEG, we use the ActiChem system with 21 electrodes placed over, according to the 1020 systems. Electrodes were placed between FNIRS fibers. We also acquired auxiliary channels to measure physiological confounds that we extracted from the data later on. Mushtaq from my lab, went to Barbados in summer 2018, set it up the lab there, the BNS clinic that Janina created a while ago. And all participants did a go-no-go task. They perform a go-no-go task, which is an attention inhibition control task that we probably know. And we also recorded a 12-minute resting state during the simultaneous EG-FNIRS recording. So I'm presenting here the first paper we published in 2022 from this data set. I have to say this is the ERP results from the EEG. The behavioral analysis from that was the EEG recorded during the go-no-go task, of course. So the behavioral analysis revealed that previously malnourished adults had higher rate of omission errors on the task related to controls, and the ERP results also align with this result. For the EEG data, we first applied a standard pre-processing pipeline, including filtering, ocular movement artifact correction and rejection, etc. And the ERP results, for the ERP result, we focused on two main components, so the N2 and P3, which are, we know that they are, it's well known that they are associated with the go-no-go task. So we found that the N2 amplitude of the PM in the PEM group, which is the malnourished group, compared to the control group was lower. So we can see it here more clearly. So these are the results from the no-go condition and from the go condition. We'll focus on the no-go condition. So this from FZ, FCZ, and CZ. The red line is the black line is the results, the waves from the control groups, and the red line from the malnourished group. So we can see that the amplitude from the N2 is larger for the control compared to PEM group. So such findings are typically associated with impaired conflict monitoring or attention deficits. and may therefore be linked to attentional and executive function deficit that have been previously reported by Janina and her team in this cohort in childhood, and again later in middle adulthood. So there are several publications showing that. So we are now, of course, we're pursuing our data analysis on EEG during resting state and doing other kind of analysis during the task, and also for FNIRS data. This is another example of what we're doing. So we are done with the pre-processing of resting state data. These are the pre-processing steps, which are quite standard. And we are also applying, we're also looking at functional brain networks and cerebral network topology using person correlations and graph theoretical approach respectively. Okay, so these are preliminary results showing functional connectivity matrices, here on the left for the malnutrition group and on the right for the control group. If we look at the subtraction between those two matrices, so malnutrition minus control, we get this matrix here, down there. And we can see that the malnutrition group seems to present stronger functional connectivity in several brain regions. However, these are really preliminary, so we need to investigate this further and do some statistics, of course, and to understand what we're looking at. We also need to do we're also applying theoretical graph theoretical approach, as I said earlier. With Pedro's teams, we're working on a fuse FNIRS EEG analysis to make the most of the richness of this multimodal signal. So hopefully, or maybe I will present these other results at the next GBC meeting. So thank you very much.
Thank you, Anne. Thank you so much. We're going to postpone questions to the end?
Yes, please, that would be better.
Okay. The next presenter is Arielle Rabinowitz, who is also a graduate student and ran the behavioral portion of the 2018 follow-up study in the population. Today, she's going to speak about the intergenerational sample and DNA methylation changes. Arielle?
Can you see my screen? Yes, we can.
Yeah.
Okay, so hi everyone. I'm going to make this quick because I know we're really short on time. So I'll be talking about intergenerational effects of early malnutrition on attention and executive control, DNA methylation signatures. I worked on this analysis with a number of other people, namely the group at Mount Sinai who did all of the epigenetic processing. So I'll just give you a little bit of background on behavioral and cognitive outcomes of early malnutrition. 22% of children under the age of 5 suffer from undernutrition worldwide.
Sorry to interrupt you. Your slides are not moving. It's still on the first slide.
Okay.
Can you start the slideshow from the beginning?
I did.
Resume slideshow. Okay, can you just click on the individual slides? We can then see them.
Yeah, I can do that. It's fine. So I'll just do this because I don't know why it's not doing full screen. But anyway, sorry. So Early malnutrition has been linked with attention and executive control problems in childhood and adulthood, but studies of intergenerational effects are limited. There are animal models that have confirmed increased distractibility on attentional tasks, as well as cognitive rigidity and associated alterations in prefrontal cortex. So this project was actually a follow-up on a study that was done in 2016, and that was published already. So that looked at the epigenetic signatures in genes in the G1 group. So that was the group that was affected by malnutrition. However, this follow-up study is documenting the persistence of attention deficits in the well-nourished G2 offspring, and also to identify the altered epigenetic signatures in genes of the G2 offspring associated with parental history of malnutrition. So the participants, like I said, were the G2 offspring of G1 parents with histories of early childhood malnutrition, or parents from the healthy control group. The mean age was 19.7 years, and the range was between 16 and 31 years. The measures that were conducted were a 30-item Conners Adult Attention Rating Scale Self-Report Questionnaire, or the CARS SSV, and the CARS C scale, or the total ADHD symptoms, was used in this report. The epigenetics were generated using Infinium human methylation 450 bead chip. About 483,000 CPG sites genome-wide were surveyed. EWAS was performed on a subset of the 74 participants with DNA methylation profiles generated for 44 of the offspring of the PEM group and 30 offspring of the control group. The analysis that we did were multivariate regression models of the CARS values using methylation, beta values, age, gender, blood cell counts, parental income, occupation, and malnutrition history as independent variables. And then in terms of the epigenetics, the differentially methylated regions were identified using a 1 kilobyte sliding window analysis. So I'll just quickly go through the results and then we can move on. So this shows the results of the mixed model. So early malnutrition, as you can see, predicts ADHD symptoms in the next generation. So the graph shows the G1 and the G2 findings with the mean CARS T scores. So we had actually 3 different subscales that were significant, but particularly the ADHD symptoms. And then in terms of the EWAS results, so this is a Manhattan plot showing that 183 DMRs were significantly associated with ADHD symptoms. So that was, again, the CARS C subscale in the 74 G2 offspring. In particular, DPPA5, HOXB6, and HLA-DPA1, primary genomic loci where DNA methylation levels are modified in the G2 offspring and were associated with ADHD symptoms. So the most significant enrichment are observed in regulation of immune function. So overall, in conclusion, moderate to severe protein energy malnutrition confined to the first year of life is associated with lasting behavioral and epigenetic changes in the next generation who are not exposed to malnutrition. Future research will focus on brain changes in the next generation associated with these behavioral and epigenetic changes. Thank you.
Okay, thank you so much, Arielle, for your presentation. And everyone, if you have any questions related to Professor Janina and her panel, you can ask now. If you want to drop it in the chat box.
Oh, I still have a presentation.
Oh, yeah. Okay.
Okay, I have a very quick presentation. I don't know whether I'm going to be able to get the slides to work, but Can we try it?
Okay. Can you please
Yeah, I
stop
have
sharing your
them.
screen, Arielle?
Yes, yes, yes.
Okay, thank you.
Yep.
That's great. Okay, so can we go to the next slide? So there are, you know, other children with malnutrition and children generally from low resource areas suffer from different kinds of adversities, not only malnutrition. And so we, in the Barbados Nutrition Study, fortunately also collected extensive data on other adversities. And our next step really will be to expand our models to also include these other adversities. And I'd like to just give you a little bit of preliminary data on this. So this would include an assessment of childhood standard of living or poverty, which we assessed at 3 different time points, ranging from all the school years, ranging from age 5 through 18. For the purposes of this discussion, we focused on the 5 to 11 data. We also have data on physical and emotional neglect, which was collected using the standard childhood trauma questionnaire that is used broadly throughout the world. And this data was recorded retrospectively at age 40 to 45, and it's a self-report of exposure to physical and emotional abuse and neglect in childhood. And finally, we also collected prospective data on maternal depression at 3 time points throughout childhood and adolescence. And we previously published, you know, a number of papers on maternal depression in this population. And these now constitute other adversities that I'd like to just briefly discuss. Next slide. Okay, so what we did was, in each instance, in order to try to make the data somewhat comparable, we undertook data reduction using principal component analysis, and these are the factors. Again, standard of living, physical and emotional neglect, which was the most significant factor from the CTQ that impacted outcome, and also maternal depression. Next slide. So the prevalence of childhood adversities by malnutrition history is shown here, and it's not surprising. If you look at 0, meaning 0 exposure to any of the 3, meaning any of the 3 adversities, you see that the control group is the only group that has 0 exposure to any other adversities. Both the malnourished and the control groups had exposure to 1 or 2 or 3 adversities. So what this indicates is a total of 1 additional adversity, a total of 2 additional adversities, and a total of 3 additional adversities. So again, if you look at the 3 adversities section, you see that the malnourished group obviously has a greater preponderance of, or greater likelihood of having all 3 exposures. Next slide. This is just a quick overview. If we only look at malnutrition and we control for the other 3 adversities, you find that, again, there remain significant differences between the PEM and the control groups, both on the composite MOCA score and the subcategories, the various subscales, and the same on the MMSE, which is the other scale that is used to look at cognitive decline in adulthood. And again, these individuals were 45 to 51 years of age when we looked at these measures. Next slide. Now, what's really interesting is when you look at the relationship between the control and the PEM group, adding in the various additional adversities, a very interesting finding happens. What you see is that if you add 1 adversity, again, you see that the, actually, let me back off for a minute. The control, the scores here on the MOCA represent 30, represents a perfect score, okay? So, and if you have under 26, you have a clinical case of cognitive decline or early dementia. So what you see here is that in the group that has no exposure, the controls, they are all, none of them have dementia, and they're all pretty close to the 30-point mark. When you have one or two exposures, you see that there are significant differences between the controls and the malnourished being substantially, significantly lower than the control scores, meaning that there is, again, a predisposition toward accelerated cognitive decline. But what's the most interesting is that when you get to 3 additional adversities, you see that there is now no difference between the control and the PEM group, and they are all below the 26 point. The mean and the median scores are below 26, meaning that all of the individuals, whether they are from the control group with no prior malnutrition or they are from the protein energy malnourished group, are all at risk for accelerated cognitive decline. And this is very interesting and actually addresses the one of the questions that was raised earlier about whether you can use some of these findings to distinguish people clinically. And the answer is yes, it appears that you can. But what's also very interesting is that this suggests that there are also brain changes in the control group, which is perhaps why Jorge didn't find, clinically significant ability to be able to separate the groups out. And that's because the match control group also was exposed to these other adversities, and the other adversities independent of the malnutrition also seem to have a very significant effect. We replicated this finding also on the MMSE, which I will not show right now. Next slide. But I will show you the soft neurologic signs that Fuleah talked about a few minutes ago. And I think you can see the same thing here. The soft neurologic signs basically are a series of fine motor skills that are tested in the dominant and non-dominant hands. And this is the total amount of time that it took for individuals to complete the testing. And what you can see is exactly the same thing, which is that there's a trend toward a difference with one exposure. With 2 exposures to 2 adversities, there is a significant difference between the controls and the malnourished group, with the malnourished group having slower scores. And finally, when all 3 adversities are included, what you see is that there's absolutely no difference, and that the control group is also showing deficits that are virtually the same as the deficits seen in the previously malnourished group. So this is very exciting. Next slide. So the summary and next steps. Early malnutrition limited to age 0 to 1 appears to be associated with accelerated cognitive decline at 45 to 51 years of age. There have been some other studies, such as the Dutch famine studies and Chinese famine studies, that have suggested similar findings, but the actual data documenting the early life condition, nutritional condition, is clearly better defined in this sample, in the Barbados sample. Second, the additional childhood adversities, poverty, neglect, and maternal depression have little or no additional impact when you're only looking at the malnourished group. So it appears that the early malnutrition is intimately connected to and associated with these other variables, but in fact it is the dominant variable that impacts brain development. In controls, however, you see the opposite. You see that when 1 or 2 adversities are presented, there's very little effect on cognitive decline, but controls, in contrast with exposure to 3 childhood adversities, did have accelerated cognitive decline with MoCA and MMSE values similar to those observed in the PEM group. So what we are planning is to expand our models, our future studies, to also include the wider range of childhood adversities that we and many others have also talked about as affecting the brain development of children in low resource settings. Next slide. Okay, so this is again the model. This is a sample model, and it's a model that was first presented in 1970 by Herbert Birch, who was my mentor at that time. And it does show, you know, the various ways in which the poverty malnutrition cycle continues itself intergenerationally and can also affect the next generation, and the interaction of the various adversities that we observe in these populations. Next slide.
Okay, Professor, sorry to interrupt you. You have 2 minutes for your talk.
I'm done. I want to thank everyone who has participated, you know, and collaborated on this study. In particular, you know, we have worked for years very closely with Pedro, with Maria, and now with Ann Gallagher, and many others, you know, who have contributed extensively, you know, to our work. So it takes a village. Thank you.
Okay, thank you so much, Professor Janina, for your presentation, and to the whole panel. Thank you so much. Actually, we are short on time, so I would suggest that if you have any questions related to Professor Janina or her panel, you can drop them in the chat box, and every presenter, they can try to answer you there. So moving ahead, now we have our next presenter, Dr. Faisal Mushtaq, and he's going to talk on city-scale neuroscience. So over to you, Dr. Faisal.
Okay, hello everyone. So first challenge, can you see my screen?
Yes.
Oh, yes, we can.
Perfect. Okay, great. So I'm going to tell you a bit about some of our plans for some work happening in the city of Bradford, which is right next door to Leeds in the UK, which is where I'm actually sat right now whilst I give this presentation. So some context, the Born in Bradford Longitudinal Birth Cohort Study is a project that was set up about 15 years ago. It's been following about 13,500 children from pregnancy, and those children are now starting the sort of transition towards adolescence. And this project, I guess, similar to some of the work you've just heard about now, well, was set up for a particular reason. And in this case, it was around maternal health inequalities, educational health inequalities, and socioeconomic sort of inequalities more generally across the city of Bradford relative to the rest of the UK. And you see some of the statistics on the side there. I won't go into any of those details right now. So we've been doing this work for about 15 years. And when I say we, again, in the last talk, takes a village. There's a sort of cast of thousands who have been involved in this project at one point or another. And recently been successful in securing some funding to continue tracking these children through the sort of transition to adolescence over the next 7 years, well, next 6 years now. We've been doing this for the last year, across a range of our existing sort of measures. And I'll give you a bit of an overview. I won't go into any detail about any of this, but you can see we've got a huge, rich sort of data set already collected, and we'll continue to collect those sort of measures as we go along. But now this is a Global Brain Consortium, and I guess you might be looking at some of this and thinking, okay, that's fantastic, but where is the neuro data? And the answer is we don't have any. So the ambition, sort of my life's mission for the next sort of few years, is to address that problem. As some of you have probably heard me talk about this over the last sort of year or so. I've been trying to develop these sort of plans, and I'll tell you more about where we're at now. So in terms of addressing the sort of problems, we've identified that effectively EEG is really the only sort of viable sort of solution here to do this at the sort of type of scale that we want to do. And then the EEG will be of interest in and of itself, but more importantly, how that links into all of the data sets that we already have on these children. The problem we have when we're doing this, and I'm sure there's a lot of lessons I can learn from people here on this sort of in this conference, is a problem of scalability. So we have about 24,000 children. I mentioned 13,000 because that's the original birth cohort study, but we've now expanded that out to all these children in the sort of secondary school. So that's the children we've been following and their classmates across the sort of city of Bradford. So we have about 24,000 roughly. And the problem is that this type of EEG that I'm very familiar with, laboratory-based sort of EEG, just doesn't scale. You just can't collect 2 time points, at least not in a way that we can sort of resource sort of a constrained sort of environment that we are in the UK with the research funding. It's just not possible to do that at 24,000 participants, lab-based sort of studies. At best, we'll get a snapshot. We'll probably get about sort of 1,000 sort of participants at 2 time points over the next sort of 5 years. That's our sort of plans. So that gives us a bit of a snapshot. We'll get some detailed sort of examination of neural sort of function with that. But we've also been looking at ways of addressing a sort of a larger sort of cohort to increase our sort of sample size and get more representation from across our cohort. And one of the ways we've been doing this is not particularly sort of revolutionary. This has been going into schools and setting up pop-up laboratories. That's one of our plans. So we just recently, we did this actually in the summer. It's been a little while now. So you see some pictures here from our team. And I would say thanks to sort of Sid for providing some of the material for some of that. I won't share the information here on that because it's just been pre-processed and hasn't been analyzed just yet. But what I want to tell you about is a third approach, a way that we want to try and collect data from everybody. And I want you to be able to, I would like people to throw stones at this approach and tell me about all of the problems so we can try to address those, because I realize this is a very ambitious sort of plan. And I would really like to get people sort of input on what we're trying to do here. So how on earth are we going to do this? So we think the answer might lie in low-cost commercial EEG. You've heard some of this sort of being mentioned by Mike earlier on. And no doubt you've come across the company Emotive, who makes some of the most widely sort of sold commercial EEG systems. And they came out with a paper last year, which I found really interesting. So they have a new platform called Emotive Labs. It's a sort of online platform where people who've purchased these systems can go and log in, and you can remotely acquire data in return for sort of a remuneration. So they demonstrated the proof of concept that this could work with 20 odd sort of participants. And they've also built a sort of a bit of sort of platform around quality control as part of that process within this remote data collection. So I thought, okay, well, maybe we can take this type of approach. Perhaps we can implement that in a school environment in a slightly more controlled way. There's just some proof of concept data there. For those of you who haven't seen the Emotive system, you'll see I'll just quickly flick through that in the interest of time. This is self-application. It's relatively straightforward. But there, of course, there are compromises in terms of channels. There are compromises probably in sort of signal quality. And those are the things that we need to be explicit about. So we think there's a way of doing this. And the solution that we think we're going to go with is a sort of a semi-supervised peer application of EEG. So I'll tell you a bit more about what that means. I'll just quickly wrap up what we're planning on doing here. So we are still going to do our lab-based data collection, deep dive with a couple of hours and sort of high density sort of EEG. We'll be going into the schools, setting up laboratories and sports halls. We'll probably be able to collect about 4,000 participants, our calculations tell us. And then we think we have an approach that will allow us to collect thousands of participants at scale. So, but there are question marks, of course, over how feasible that is and what the sort of trade-offs are in sort of signal quality. So, where are we? Yes. So what does the semi-supervised peer application look like? Well, effectively, we go into a classroom, we pair up participants. We get typically a UK classroom is the size of about 30 top end, 30 sort of students. We pair them up and we work through a sort of a protocol, a sort of workshop is what we're calling this. I'll just quickly go through here. A workshop like this, where we introduce them to sort of neuroscience, we introduce them to EEG, and then we get them to apply the EEG headsets on one another. They take turns to be experimenter and participant, and then we sort of exchange the sort of equipment over, of course, cleaning it in between. So there are some questions. So I have many questions about the feasibility of this, and I'm sure you will have questions too. And this is what we're now working on. We're trying to sort of address some of these questions, understand what the sort of consequences of doing this type of work are. So, you know, are teenagers really going to be able to apply it? Are we going to be able to follow the task instructions? When you're collecting data simultaneously from 15 participants, what are the consequences? What are the environmental noise? There's electrical interference. What about people who traditionally historically have been left out when it comes to EEG research? So people sort of with thick, curly hair, sort of a Afro-Caribbean sort of background. We know this, that sort of a very less sort of a representation in the EEG sort of literature of participants with those characteristics of their hair. So how do we address those sort of problems? And then the big one is quality control. How are we going to address that? And of course, the question that we have is this sort of signal quality sample size trade-off going to be worth the effort? So these are all the questions we're trying to grapple with right now. So just quickly being through that. We have got some processes in place where we're trying to mitigate sort of for these sort of quality control issues. So on the left-hand side, you see that Emotive, as part of their Emotive Labs sort of a platform, have a sort of signal quality, automated sort of signal quality sort of checking built in, which is good. On the downside, it's a proprietary sort of algorithm, so we don't have the secret sauce. We don't understand how that sort of works. What we do know is it does seem to sort of give alerts in a timely fashion. So it seems to have some sort of face validity to it. At this point, I should also say thanks to Emotive for giving us a free team license. That's a disclosure there for allowing us to be able to carry out this proof of concept work. And we've also been looking at, thanks to advice from Bosch and Pedro, looking at sort of tracking electrode positions as well, sort of see how much variance there is in application from one participant to the other, and also testing whether there's a shift from the start of the session to the end of that as well. So we've just started piloting that. And then ultimately, there's no substitute. You can have all the automated sort of approaches, but you really need sort of people with expertise in understanding the sort of signal. So we've developed an approach now where we have trained EEG research assistants who give a green light for recording, and will continually monitor this sort of via screens in the sort of room. So you just see a picture here. So this is very, very early in the life of this model for data collection. These are pictures literally from yesterday, which we tested in, we tested in an undergraduate sort of population. Next week, we're going into a school, testing this on 25 sort of children. So it's still very, very early. We still don't know sort of whether we'll be able to fully, adequately address all the questions. But this is where I thought it was a timely point for me to present this to you and hear your thoughts about it. And then there's sort of, I've talked about the challenges of doing this work, but there are also opportunities when you're engaging with that many young children from across the whole city. There's an opportunity for us to, sort of, I guess, sort of enthuse the next generation around neuroscience, around data science, around sort of engineering, neurotechnology, and sort of digital sort of skills, etc. And that's what we're also doing as part of this sort of program. So when we go into a school, we take out time out of their day to collect the data, but in return, we come back and we run a sort of a data science type sort of a workshop, which gives them a bit of an insight into the sort of scientific process, and also hopefully gives them some skills that are aligned with the curriculum. So we've been doing some of that historically. This is a picture where this is actually from a primary school where we did some pilot work with our pop-up laboratory, where we've got, that's one of our RAs, Layla, who's introducing neurons and modeling those using pipe cleaners. So we've got education sort of embedded into our sort of program. So as I mentioned, we're still very early, but we've got sort of ambitions to scale this up very quickly, this particular approach. We will be piloting next week, and also expanding the sort of types of sort of tasks that we might do. And thanks very much to Sarah, who's offered an auditory entrainment task that we'll sort of pilot to see what's the extent, how much can we push this. So we've got resting state right now. Can we do ERPs? Can we look at sort of videos, etc.? So we'll be exploring all of that very soon. And then we'll sort of collect data from the whole school before the end of the academic year, this one school that we are partnering with. And then I hope to show you the results, good or bad, at OHBM in Montreal later this sort of summer. So I will wrap up there and hopefully give some time back to this program and take any questions either now or on sort of at the end, whichever is appropriate. So let me stop sharing now.
Okay, thank you so much, Dr. Faisal, for your informative presentation. And we are glad to know that you had a lot of questions and you're open to suggestions as well. So everyone, if you have any questions, you can drop them in the chat box as we are short on time. And we can move ahead to our next professor, Guisip Chirenza. Pardon me if I am pronouncing it wrong. So he's going to talk with Bosch on the clinical subtypes of developmental dyslexia have a distinct neuropsychological pattern. So up to you.
I will explain briefly this work. This work is a study we did on different types of dyslexia. This is a database that Professor Chirenza has been.
Hello,
Yes?
Bosch. Sorry to interrupt you. Your
Yes?
screen is okay. Now it's okay. Carry
Can
on.
you
Thank
see?
you. Yes, now we can see.
Okay. So this database is a database of different types of subtypes of dyslexia that Professor Chirenza has recorded for many years. It has more than 300 different children in different types of dyslexia. He has this phonetic, he has this eidetic, and he has a mixed group, which is a mixed group is the type of more severe type of dyslexia. What we did in this work, so let me see if I can move this. Okay. So the dyslexia children has been classified according to Boder's criteria classification system. This is a system that was developed in the 70s that Professor Chirenza he validated and developed a version for the Italian children. And this system classifies the children in this type of dyslexia, but this is a behavioral system. There was not any neurophysiological validation of this system. So what we are doing here is for the first time trying to validate the subtypes of dyslexia described by Boder using the EEG. So he has the description of the different type of what Boder, in which Boder was based to distinguish different types of dyslexia. I'm going to go fast through this. Maybe if Giuseppe can join later, he will like to talk about this. And so this is all, and these are the differences. So the different types, and then I will come here. What we are showing here is a very nice example of the usefulness of the new harmonized QEEG norms that we developed last year under the boundaries of the GBC. On one hand, we have the traditional Cuban normative database that we have been using since the 90s. We have the Cuban methodology of QEEG for the scalp and the sources. And we have now that we developed last year, the new harmonized QEEG. What is what we have new with this new harmonized QEEG? We have first, instead of the Cuban database, we collected the multinational database of more than 1,600 subjects from 9 different countries. There was developed an algorithm procedure for batch correction, so for harmonizing the data coming from different devices and culture and databases. And third, we included a new type norms. For the first time, we included norms for the full cross spectra. Previously, we only have norms for the log spectra, and then we wanted to know how better is this new QEEG norm compared to our previous methodology. Since we have this, we were able to use, because we didn't have control group, we were able to use part of the 100 children from the New York database collected by Dr. Leslie Pritchett and Roy Young, that are part of the normative group. So we use them as a control group from this. The main reason we did that is because now we have this procedure to harmonize between different devices, but the other reason is that these children were recorded with the same or a similar device that the one used by Chiarenza to record his dyslexia children, and they were recorded more or less during the same time. So we have this number of children. We have 290 subjects. So you can see here, 185 dysphonetic. We have 48 in the mixed. This is a very rare group, and we have 18. This is all the EEG specifications, and then we apply the traditional QEEG normative. So we compare the traditional QEEG norms with the new harmonized QEEG. We have the harmonized on the other hand. And then what we did to find the classification power of the two methods for classifying the different type of dyslexia was to use a methodology that we developed that is a sparse and stable classification measurement that is based on the use. So it is based on repetitive, random repetition. So randomly repeating the classification procedure with different subsamples, and then to obtain the classifiers that participate in the classification equation at least in 50% of the times they were classified. And so this guarantee that the classifiers are robust, are stable. And also we use the elastic neck technique that produces a sparse solution. So the number of variable participating in the regression equation are few. And also we apply a robust technique for the ROC curve to measure the area under the curve of the ROC curve. So these are the results that we obtain with the all that QEEG normative database, in which we only use the log spectra. As you see, there is a good separation of the control group, but the separation between the three types, sorry?
I
don't know if this is good.
I think Professor Giuseppe is here.
Professor Giuseppe is here.
Okay. Giuseppe, do you want me to finish this part and then you can talk the first part?
Hello?
Hello. I already started. Do you want me to finish this part and then you explain the first part?
Hello?
Yeah, I think he's not listening.
Yeah, I think he may have some internet connection issues. So
yes. So I will continue. I will finish this part.
Okay.
With the all QEEG methodology, we were not able to separate the three dyslexia types. So you see the area under the curve was really very poor. The good thing about this is that we did the same with the new harmonized norms, but under the same condition as the all normative. So we didn't use the harmonization procedure, and we only used the log spectra. And the results that we obtained with the new norms were very similar to the ones that we obtained with the old methodology. This is very good because this gives us confidence on what we are doing. However, when we use the classification with the new QEEG norm using all the features, including the harmonization procedure and using the full cross spectra matrix, we obtain a very high classification between the three groups of dyslexia, and the control group was 100% separated from the different three types of dyslexia, and we had a very high classification also among the three groups. So from group 1 and 2 was 85, 94, and 78. So they use the inclusion of the cross spectra in the classification. It improved very much the classification power of the EEG to differentiate between the different groups. These are the classifiers that we obtained from the new norms. As you can see, they are correlations among variables. So the majority of the variables that participated as classifiers are cross correlations, cross spectra, not precisely the log spectra. Only two variables part of the log spectra, which is T6, participated in the classification power. So this is the greatest power of the new classification, the new methodology that allows to include the inclusion of the full cross spectrum. We did another comparison, and what we are plotting here is the imaginary part and the real part of the cross spectra, the thick cross spectra. So, you know, the cross spectra is a complex number. The real part means the amplitude, and the imaginary part refers to the phase. So the phase difference between them. If we look at the control group, they are all very symmetrically distributed around 0. However, if we look at the dysphonetic and the dysidetic group and the mixed group, they show very different patterns. If we look at the dysphonetic and the mixed group, they look very similar. And this is what I was saying at the beginning, that the mixed group is a kind of more severe dysphonetic. And this was also a very nice result, that they look so similar here, and not the dysidetic that shows a different tendency or a different displacement in this axis. This opens a new avenue for studying this, because we can, from here, we can see which are mainly the main differences between the groups. Is it in the amplitude of the cross spectra, or is in the different phase? So this is the next step we are going to do. I don't know if Giuseppe is around there, if he wants to. I will stop sharing my screen now, and let's see if Giuseppe is here.
And okay, again, sorry for being late, but in 1973, Elena Boder, a pediatric neurologist, published in the journal Developmental Medicine and Child Neurology a reading and a writing test, with which she claimed to be able to identify, by directly observing reading and spelling errors, different subtypes of dyslexia. Subsequently, inspired by this test, I developed a semi-automatic and a computerized version for the Italian readers to demonstrate the existence of these subtypes also in Italian-speaking dyslexic. Italian language, by its nature, is defined as transparent. That there is a neurophysiological difference between dyslexics and normals is a fact now established by many researchers. What remains to be demonstrated is the existence of different neurophysiological patterns among subtypes of dyslexia. So the aims of this presentation is to describe the broader classification system of dyslexia, to validate the classification system of Boder through the harmonized QEEG cross-spectra norms, and to identify the possible neurophysiological patterns of dyslexia subtypes that now you already know. So the Boder system is based on a general model of network cognitive brain functions. This is the most important aspect, the network. According to this model, the cognitive brain functions are represented in a widely distributed neural networks, interactive and overlapping. Reading may be said to be essentially a two-channel function requiring the perfect dynamic interplay of intact visual gestalt and analytic auditory function, and integration of both peripheral and central processes. The key words important for this study is this dynamic interplay and integration of processes at the level of this network. The normal reader recognize familiar words which constitute their sight vocabulary through the visual channel as instantaneous visual gestalt of whole words without having to discriminate individual letters or component syllables. They read the familiar words on sight or visualize them. The normal reader recognizes unfamiliar words through the auditory channel by a process of phonetic analysis and synthesis. These are the mechanisms for the normal readers, how the children, they learn to read, to write, by learning by sight or by syllabicating the words. The two channels are active and they shift from one to another according to the words are familiar or unfamiliar. In dyslexic children, this normal reading process is dissociated. The normal dynamic network interplay of gestalt and analytic synthetic processes is disrupted. Dyslexic child read and spell differently from normal readers, both qualitatively and quantitatively. Dyslexia is therefore a nosological entity defined by specific cognitive deficit patterns whose clinical characteristics depend on the dynamic interplay between cognitive tasks attributed respectively to visual gestalt and auditory analytical channels. A deficit in one of the two channels generates specific errors characteristic of the impaired channel. So just looking at the type of errors that the child makes during reading or writing, you can diagnose which subtype of dyslexia the child has. Therefore, if this is the model. When you have a strong Gestalt visual function and a weak auditory function, no, then the subtype is called dysphonetic. If you have both a strong visual and auditory function, then you have a normal child, or at least an aspecific reading retardation. On the contrary, if you have a weak Gestalt visual function, they are not able to see the letters, recognize the form of the letters, but you have a strong auditory function, then you have this diseidetic type of dyslexia. So the brain in these children, because of this dynamic interplay is disrupted, the dyslexic child uses the most active channel. And so if he has this disturbance in recognizing the form of the letters, he will be a slow reader because it activates immediately his analytical function, and then he reads syllabicating all of the words, even the familiar ones. The hypothesis was that to describe this dynamic interplay between the various reading modalities at the...
Sorry to interrupt you, Professor. You have 2 minutes left for your talk.
Yes,
Thank you.
This is the end. The hypothesis was to describe this dynamic interplay with the new harmonized multinational QEEG norms. These are the database consists of 290 subjects, and this is the percentage of dysphonetic, 61, 50 is the reading retardation, is 48 subjects, the mixed group 16%, and diseidetic 18 subjects, 62%. So this is the data acquisition. Okay, I think that here already Bosch has explained the value, the method that we have applied.
Yes, I started from here.
Okay. Thank you so much, Professor Giuseppe and Professor Bosch, for your talk. And we are going to end our session here. Thank you, everyone who participated and who presented today. It was a very informative session, and we can have a Q&A session and discussion session tomorrow for our day 2. So see you all tomorrow. Till then, take care, and thank you.
Thank you.